Site-specific self-read genes as tools for the control and genetic engineering of natural populations. Site-specific self-read genes recognize a specific nucleotide sequence within the cell that synthesized them and then insert themselves in this DNA target. The repairing system of the cell then leads to the encoding of the self-read gene in the chromosome sequence. We distinguish three types of site-specific self-read genes, which are homing endonuclease genes, group 2 introns, and some line-like transposable elements. Only the example of HEGs will be exposed due to a simplest mechanism, but the two others present diverse applications too. In this video, we will show you different ways to genetically engineer those genes in order to target new sequences, allowing the manipulation of natural populations. The goal of the construct is to eradicate some species potentially harmful to human condition, particularly species that cause and transmit disease, but also species that reduce agricultural outputs. The main advantages of those constructs compared to other techniques currently used is their stability and reversibility. Indeed, transposable elements and bacterial symbiotes show a lot of problems due to their lack of stability and thus cannot be used for such application. The whole process is based on the recognition of new targets, giving new possibility in functional genomic and gene therapy.
We are going to discuss now the construction of site-specific selfish gene, that is, how they work and ways of increasing its functionality. HAGs are selfish or parasitic genes that can spread through population owing to their super-Mendelian inheritance. They encode an enzyme that can recognize and cleave a 20-30 base pair sequence found on chromosome not containing a copy of the HIG. They are inserted on the gene we want to knock out. The HIG itself is inserted in the middle of its own recognition sequence, and so the chromosomes carrying the HIG are protected from being cut. When this gene is inserted, the chromosome from which it comes is broken, called HEG-. It will be repaired by the cell recombinational repair system, which uses the intact HIG homologue as a template. After that, we will have an homozygote. In wildness, they don't disrupt essential genes because they are associated with self-splitting group 1 introns or inteins, but that is not the case of engineered ones. The main features of site-specific selfish gene are the evolutionary stability, the reversibility, and wide applicability. A key feature of this construction is that it is evolutionary stable, in the sense that the mutant forms are likely to arise as it spreads through a population will be selected again and lost. The construct is fully reversible. If one target a gene that, when knocked out, is strongly deleterious, then there will be a strong selection in favor of resistant alleles. We can use this technique in most eukaryotes and also in haploids. In haploids, it is only needed at least a short diploid phase in which one could target a gene encoding for the protein needed. Another important thing to be taken into account is the number of generations needed for the population to become homozygote. Depending on the gene affected, the number of generations vary until a population is mostly homozygote. Increasing the load is a way of doing it quicker. There are two main methods that are targeting alternative loci and targeting multiple loci. For many species, one might want to control the number. In that case, it has to be taken into account that the rate of population growth is determined most by female productivity than male. For that reason, when targeting alternative loci, a possible target gene will be one that, when knocked out, kills only females. On the other hand, we can also target multiple loci simultaneously. By statistics, the more loci you target, the quicker the population is driven to extinct.
The most important application of this engineering tool is the population-wide knockout. It consists on the knockout or elimination of genes present in noxious populations, for example for humans, so that we can obtain an non-noxious population, as the genes are responsible for the deleterious feature. As seen in the figure, knockout of genes consists on the insertion of a gene inside the open reading frame of the gene we want to silence, so that it is to interrupt and no more functional. The genes affected are essential for the toxicity of the population, but not to live, so the population is not lost. The target gene is chosen such that the knockout mutation has little phenotypic effect in heterozygotes, but is severely deleterious when homozygotes, which means that the knockout is recessive. We introduce the mutation, as explained before, so that we obtain an heterozygote, and the population is still toxic for humans. We have to wait many generations until the individuals are homozygotes for the mutation, which is called spread to fixation. At that moment, we have modified the population in such a way that it is no more harmful for us. Knockout mutations destroying the function of the toxic gene are sure to arise, and these mutations will spread at the expense of the toxic gene. Unless one can achieve quite high release frequencies, such an approach is probably limited to modifying a population rather than eradicating it. One example of population is the mosquito that transmits malaria. If the knockout is not too harmful, then the HEG, which is homing endonuclease gene, will spread to fixation, and this may have little or no effect on population numbers. Many genes could be knocked out in this way. Such an approach also has the advantage that resistant genotypes will be less strongly selected.
As been said before, the knockout of some of these genes can be harmful for the population if we are talking about an essential gene. For that reason, there are other applications for the HGs to avoid the possible harmful effect. We will take now into account the genetic engineering of populations. When we talk about white population gene replacements, we suggest the possibility of just modifying some aspects of our essential gene instead of knocking it out and therefore provoking the death of the organism. We can take into account the example of the mosquito. Let us imagine that the gene in charge of transmitting malaria is essential for its life. We would like to modify this gene in such a way that it would be lethal for the production of malaria but has no effect on the mosquito. We can then design an HG that blocks the unwanted sequence but leaves the desired one. If we release these mosquitoes carrying the HG and the resistant allele, this later one will predominate quickly, as can be seen in the figure. Another alternative is the white population transformation. This is driving a novel gene into the genome of a population. This can be done with our HEG in two different ways. First one, the novel goes linked to an engineered HEG targeting a specific region of the genome. Another option is having the engineered HEG to target an essential gene, and on the other hand, our novel joined to a resistant allele, for example, using an inversion. This could allow the binding of multiple genes as well. However, we have to take into account that if the novel gene is harmful for the population, the construct will not be evolutionarily stable and would disappear along the generations.
In some mosquitoes, the Y chromosome causes the X chromosome to break during the first meiotic division, and thus show transmission ratio distortion. This molecular mechanism is not yet known, but there's a strategy. We can insert on the Y chromosome one or more endonuclease genes that recognize and cut the specific sequence on X chromosome under the control of premeiotic specific promoters. During spermatogenesis, the X chromosome would be cut, so it's not more an appropriate template for repair. The Y chromosome would show transmission ratio distortion and spread through the population. If the transmission ratio distortion is extreme, the population would eradicate. The release of relatively few engineered individuals will drive the population manipulation. Often this is an advantage. If we want to eradicate only one population and leave others, we cannot use inoculative methods, but we can use inundative strategies, like the release of sterile males. In these strategies, we can use HEGs if they cause dominant female lethality or sterility. If HEG was engineered to be constitutively active in all tissues, the organism would be converted to an homozygote. Females with HEG would be dead or sterile, and male would pass on the HEG to the next generation. If HEG is not perfectly efficient, it would slowly disappear from the population. But before doing it, it can cause a substantial load. So by changing the promoter, the threat posed by rare emigrants to neighboring population can be avoided. The use of those engineered HEGs would be more efficient than the release of sterile males, allowing either fewer individuals to be released or larger population to be targeted.